The overall goal of the following experiment is to record subcellular events during early embryogenesis in Caenorhabditis elegans. This is achieved by using transgenic strains with multiple fluorescently labeled proteins that allow the visualization of DNA as well as subsets of vesicles in the early embryo. Specific organelles are labeled via microinjection of fluorescent dyes such as lysotracker or mitotracker into the gonad of the adult worm. Next, embryos are cut out of adult worms and observed with a confocal microscope to obtain time-lapse recordings of the movements of the labeled proteins and organelles. Ultimately, the protocol reveals interesting movement of lysosomes and GFP-tagged vesicles during early development in C. elegans.
My name is Connie Hedjar, and today I'll be showing you how we perform time-lapse microscopy in our lab, the Boyd Lab, at the University of Alabama in Huntsville.
For this experiment, grow nematodes on NGM agar plates seeded with an OP50 bacterial lawn at 25 degrees Celsius. The day before the experiment, pick at least 40 L4 larvae onto seeded plates and incubate them overnight at 25 degrees Celsius. These worms will be young adults for the experiment. Make an injection pad by sandwiching 2 drops of 2% agarose in water from a Pasteur pipette between 2 cover glass slips and applying pressure to shape the agarose into a 1.5 centimeter diameter circle. Remove the top cover slip and dehydrate the pad by placing it in an 80 degrees Celsius oven for 1 hour, or allow it to sit on the bench top overnight. On the day of the injection, prepare a diluted mitotracker or lysotracker dye solution and pull injection needles from a 1.2 OD glass capillary. Backfill the needles with a diluted dye solution and store them in a dark humidified chamber. Mount injection needle onto the micromanipulator and connect the needle to a pressure regulator with a 1.2 mm internal diameter tube. Now, place 2 drops of heavy mineral oil onto the injection pad. Place the injection pad onto the microscope and lower the needle into the oil. Apply injection pressure and observe whether fluid flows out of the needle. If not, you will need to gently break the end of the needle by gently driving the tip into a small bit of broken cover glass placed on the injection pad. After the needle is flowing, proceed with the injection. Approximately 1 hour prior to viewing embryos, use a pick to transfer young adult worms into the oil drop on the injection pad. Arrange 3 to 10 worms in parallel and a little less than 1 worm length apart. It is now important to inject the worms quickly before the worms desiccate. Move the pad with worms onto the microscope stage. Focus onto the central part of the distal gonad. Then use the micromanipulator to move the tip of the needle into the same focal plane. Puncture the worm by moving the stage horizontally. When the needle tip is inside the rachis, apply pressure to fill the gonad with a dye mixture. The gonad will expand with the volume of the injection. Proceed with injecting both gonad arms of each worm. After injection, cover the worms with 0.5 milliliters of egg buffer applied using a pulled Pasteur pipette. Then allow the worms to recover for 1 to 2 hours at room temperature in a light-free, humidified chamber. Begin by preparing a new agarose pad for viewing the injected embryos. With the Pasteur pipette, put 3 drops of molten agarose onto a clean microscope slide and place that slide between 2 other slides wrapped in 1 layer of labeling tape. The tape controls the thickness of the pad. Next, Press the clean microscope slide down onto the agarose drops so it rests on the taped slides. Do not remove the top slide until the embryos are ready to mount. Now, drop 20 microliters of egg buffer onto the center of a cover glass and transfer the injected worms into the buffer. Under a dissecting microscope, cut the worms open near the vulva using a 26-gauge hypodermic needle. The embryos will spill out of the worm. Now remove the top slide of the imaging slide and lower the agarose pad upside down onto the cover glass with the embryos. If the embryos will be imaged over an extended period of time, seal the edges of the cover glass with petroleum jelly to prevent desiccation. Now begin imaging by locating embryos under the 10x objective. To find early-stage embryos, search for embryos that are in the process of completing maternal meiosis. Meiotic embryos can be distinguished from later embryos because they have not undergone shortening. After completing meiosis, there is a significant gap between the cell membrane and the eggshell at the anterior end of the embryo. Once an appropriately staged embryo is identified, move to a higher magnification lens for recording of embryogenesis. This embryo expresses both M-cherry histone 2b and GFP ubiquitin tagged proteins. The maternal and paternal DNA is visible via the M-cherry H2b and thus shows the stages of early development. This embryo was injected with lysotracker blue and expresses GFP ubiquitin. The images were collected using a 63x 1.4 NA lens with 488 and 555 lasers set at minimal power and maximum gain. Typically, Z-stacks are collected every 10 to 15 seconds for up to 60 time points before photobleaching occurs. For this time-lapse video, the Z-stacks were collapsed into maximum projection images.
While performing time-lapse microscopy, embryos should begin pronuclear migration within a few minutes of completing meiosis II, and within 20 minutes the first cytokinesis should begin. If you observe arrested development of your embryos, try decreasing the laser intensity or use a thicker agarose pad.